I'm really honored to speak here today because my academic journey was actually starting at the Technical University of Berlin, so it's nice to be here and present now my PhD project, which I'm currently conducting at the University of Twente at the Applied Stem Cell Technology Group under the supervision of Andries van der Meer. And today I will talk about an open-top organ-on-chip platform which we developed to model the outer blood-retinal barrier. Just now a short introduction. What is actually the outer blood-retinal barrier? So we all have it, and it's lining the inner back of the human eye. And the outer blood-retinal barrier is the most important tissue to maintain the vision in human. The outer blood-retinal barrier consists of a vascular layer, which is separated by a Bruch's membrane to retinal pigmented epithelial cells. And on top of the outer blood-retinal barrier are photoreceptors resting, which will be completely supplied by the blood perfusion in the choroid. And as you can imagine, any changes within the choroid in the blood flow or reduction of the blood vessels can directly then also affect photoreceptors. And in fact, actually this happens to everyone, to us. As we are aging, we see like a reduced blood vessel network in the choroid. We see that the Bruch's membrane is actually thickening, which leads to like a male supply of photoreceptors. And until to a certain threshold, you won't be affected by this, but if you're above this threshold, it can happen that you develop symptoms, and they're like diseases like age-related macular degeneration, where in the worst case you can use actually the central vision in your vision. And what happening there is, for example, like in the most aggressive form, which is called the wet AMD, we have like vessels newly forming, penetrating the Bruch's membrane and the RPE layer, and then fluid leakage can happen, and there can complete detachment of the photoreceptor happen, and we're getting blind. Interestingly, we can pick up all these symptoms and see these changes in the clinic, but we don't know the underlying disease mechanism, and I think this is why we're all here, because we need better in vitro models to study also human diseases. And in my thesis, or my PhD project, we want to model the outer blood-retinal barrier, not including photoreceptors, but we want to model the outer blood-retinal barrier. We want to have a functional vascular network, which also maybe then can induce neovascularization. We want to have a completely hiPSC-derived model, because we also want to then in a later phase implement patient-derived cells that we really can pick up AMD disease factors in our in vitro system. And for this first, at the beginning of my PhD, I had to first develop a platform because for us there was no suitable available. And this is just a quick design of the platform. So it comes now in a well-played footprint. We have 9 individual chips which can be individually addressed. And the design is more like the classical design, also was just presented by Roger Kam. But we made some modifications. So we have in the center like a gel chamber, which is open top. So because we wanted to have a chamber that we can also afterloading like a hydrogel for making a vascular bed, that we also then have access to it and can stack up additional cells. We also eradicated in this design gel inlet channel, because we say we have open top, so we don't need additional channel to able to inject a hydrogel, which also then simplified the model. As a material for the model, we also wanted to not use PDMS anymore, and we looked for materials which are suitable for cell culture, and we decided we go for PMMA, which we micromill with CNC, so we're not using clean room facilities. In one solid block, so we have 2 sides which we are milling, and on the top side we have the reservoirs, and on the bottom side we have the microfluidics with the open top cell chamber, but we're not bonding 2 blocks together, so it's a quite leak-proof design and method, which is really reliable. And then to close off the bottom, we just simply use a commercially available qPCR tape, which also comes with the benefit these tapes are developed for imaging. So we have really highly access for imaging methods for confocal microscopy in our platform. And then just to keep it aseptic, we just use a commercial lid from a well plate, which then close off the system and then integrate it in our lab. Just a little bit for the experimental setup, then also to model the outer blood-retinal barrier. So we are starting with injecting like a cell-laden hydrogel, which consists of fibrin and collagen, where already like hiPSC-derived endothelial cells and vascular smooth muscle cells are integrated. So we inject it, and then within 24 to 48 hours, we already see that a complete vascular bed is forming. And then on top of this, we are adding RPE cells, which are also hiPSC-derived. And then the whole system is continuously under like on a rocking platform, so we are applying continuously over the whole time bidirectional flow. And then we come more like to now the actual model. So first we see here like in a bright field image the vascular bed which we are forming in the gel chamber. Just as an orientation on the side here, you see this open row of pillars which separates the medium channel with the gel chamber. And then here we have the chamber, and this inner ring which we see here, this is the actual open top area where we then can use for co-cultivation with the vascular bed. And you already see here now also after 5 days that we have like a completely developed vascular network also all over the sample. It's nicely interconnected. And also we can see in the openings between the pillars that vessels grow below there, because this is where actually oxygen and metabolites are coming, and then with this we perfuse the network. Here's now a fluorescent picture. It's a tile scan also of one of the complete chamber, and it's stained again with nucleus stain is in blue, and green is SETO1. And we also can see that it's nicely expressed, the SETO1, which is a tight junction marker, all over the vessels which are developed, so also indicating that we have a functional endothelial barrier in our system. In addition to this, we also had to test, can we actually grow RPE monolayer on top of the hydrogel we are using to form the vascular bed? And also we wanted to know, is the medium compatible we want to use for the co-culture with RPE and with the vascular bed compartment? So we were conducting a lot of tests, and this is now the final results. So first of all, here you see like just the co-cultivation area in our chip. We have SETO1 is a tight junction marker, which is also highly expressed by RPEs. We see that the RPEs can form a nice monolayer, and also that they are like in the phenotype which we are expecting also in vivo. Also importantly, this is just a 3D reconstruction of the same image, so where you can nicely see that we kind of create, like with this open top in the hydrogel, kind of a micro well. And we had also to adapt here the design that we don't create like kind of a ramen soup bowl, so we wanted more to have a pizza plate, because if you would have a deep ramen bowl, the cells would just clump into at the deepest point, and then you don't get like a monolayer just overgrowing the hydrogel. And with some optimization, we could manage this, and I think you all agree that this is a nice monolayer which is overgrowing the whole cultivation area. And also here, I think it's nice to see, it's just a picture I did with my smartphone. Here you can see, as we are using pigmented cells, we really have like in the center really like dark spots, and this is also like always then the first confirmation when I take the place out of the incubator, where I can see, okay, yeah, we have actually RPEs growing on top of our hydrogels. And this picture is even taken with the co-cultivation now with vascular bed and the RPEs. And if you look deeper then into the whole system, so this is now fluorescent image of the co-culture RPE with vascular bed, and the marker here in green is SRIN1, which is more specific for RPE cells, but also indicates like the maturation of retinal pigmented epithelial cells. Also nice to see here that we confirm the RPEs in the center in the co-cultivation area, so we don't see the cells all overgrowing the complete hydrogel and migrating into the hydrogel, but we also can see that vascular networks are forming, and also nicely all over the whole cultivation and also the gel area. And then in the next step, we also wanted to know, because this is really important also then for follow-up experiments, what we want to know, can we actually have a perfusible functional network in our chip? And for this, we just tested it with blood. So we wanted to see, can we perfuse our networks also in the co-culture with blood? And as you can nicely see, this is now zoomed in, that the red blood cells, they're really are following like the network structure and they're not leaking out of the networks. And also for the smaller more capillary size vessels, we also see that they're all perfusable. But then just having, again, a little closer look in fluorescent images, also just wanted to indicate that what we always see is like we have like more at the bottom of our chamber, we have like more wider vessels, but we also then see that vessels sprouting out from this, let's say, bottom network really towards the RPE cells. And as you can see here on the right-hand side, these vessels are really come in really close contact with the RPE cells. And we now think that these vessels most probably go into transdifferentiation more towards choroidal-like capillaries. We haven't confirmed this yet. We're waiting for transcriptomic data on this, but we assume that they are more going towards choroidal phenotype. And then also just a little zoom in then into the RPE monolayer, also just to show that aspirin is really highly expressed and also it's apically expressed, so also indicating first we have a nice monolayer of RPEs, but also a mature monolayer of RPEs. And then just to summarize, so I think I could show you that we have a platform which is working quite well. We also now collaborate and grow different models in our plate. And I think the feedback I always was getting, it's really user-friendly, there's no leakage, and it's quite a robust and stable platform. We can now model the outer blood-retinal barrier. We have a fully hiPSC-derived model. We have functional microvascular network, and we can make the co-culture of RPE with the vascular bed. And we now think we have a more suitable model to really look into this crosstalk between like the RPE and the choroid. The next steps, so just to maybe sort it in where we are, like in the first picture I was showing. So we are now want to go actually into the step where we try to pick up disease modeling. So we are currently like working also with our collaborators to implement patient-derived cells, specifically for wet AMD. And we also are looking into how we can induce AMD pathophysiology, for example, like neovascularization. And yeah, with this, I just want to thank everyone involved, so especially my group, which we now call the extended family, and all the collaborators involved in the project. And I'm happy to answer your questions.
Thanks. Nice talk. Just a couple of very simple questions. I couldn't quite tell from your model if you have Bruch's membrane modeled in there, or were you just putting the RPE directly on top of the vascularized fibrin gel?
Yeah, so we don't add like a synthetic membrane which will separate both the compartments, because we believe after a while, like if we mature the model long enough, the Bruch's membrane will be secreted by both compartments itself. We are testing this, we have some indications, but I didn't want to add it because it was not completely conclusive yet.
That's fine. And is there any stromal cell that you use in the vascular compartment, or is it just iPS-derived endothelial cells?
It's iPS in co-culture with vascular smooth muscle cells.
Thank you.
Hi, great talk, thank you. I have a technical question about the cell culture condition for the vascular unit. As I understood, in the outer BRB, you don't want sprout formation into the epithelium. How do you keep your vascular network from sprouting into the epithelium and also manage to maintain them in culture for long, knowing that all growth medium contain growth factors and those induce sprout formation?
Okay, so your question was, so how do we keep the network in
the
line
vessels
and not
from sprouting into the
through the RPEs?
Exactly.
Yeah, okay. So what we're doing, we first form the vascular bed, and then we supplement the media also with growth factors like VEGF. And then as soon as we see the RPEs, we take this out, and we then perfuse the network only with vascular medium. And the RPEs we keep in the RPE medium. And if we take out completely VEGF, we see that at least there's no gradient then from RPE to the vasculature. And we don't see, if we don't want to induce it, that it's penetrating the RPE monolayer.
Okay, so the second medium doesn't contain any VEGF?
No.
Okay, thank you.
Do we have any other question? Okay, well, in that case, thank you very much for your presentation.
Thank you.
And we'll move on to the next question.